give a hand for Christian Stamm from Ecolmpact.
Okay, thank you very much for the very kind introduction, Roy, and also for the very kind invitation to come here and to Waterloo and to share experiences with so many colleagues working in related fields. So as mentioned already, I would like to share some results that we obtained over the last 4 years on the issue of micropollutants. Then I would like to outline the research questions and the basic kind of research approaches and avenues that we were following over those 4 years. And then I will select some main findings that we have obtained and share them with you before I have a short outlook for finishing. I start with this picture of kind of daily life products, because what all of they share, that there are many chemicals in them. And so a large suite of chemicals is part of our daily life. And it's therefore not a surprise that at least part of those chemicals find their way actually in the water cycle through sewer systems. And what you see here on this figure is a compilation of data that we have obtained a couple of years ago, where we screened 9 typical Swiss municipal wastewater treatment plants, and we were screening for a large range of micropollutants, organic micropollutants. So we were screening for about 450 different compounds, being household chemicals, pharmaceuticals, corrosion inhibitors, you name it. And what you see here are the average numbers of the findings that we have. So on the right-hand side, this bar indicates the average number of micropollutants that we actually could detect in sewage that is discharged into the stream. So we are in the order of 140 different compounds on average that we find in treated wastewater. And what you also can see, sorry for the bad colors here, this stands actually for pharmaceuticals. So they make up a large portion of what we actually find. On the left side, what you see here, these are actually the average concentration if you sum up all of those findings in the treated wastewater. So we discharge on average something like 75 micrograms per liter of identified micropollutants into streams. What we also know from other studies, if we go for non-target screening, we can identify thousands of other peaks where there are additional compounds that are discharged into the stream. So this is the kind of the background with regard what characterizes kind of treated sewage with regard to micropollutants. So you might ask, okay, why do we care about those chemicals? And I would like to illustrate that point with one quite prominent example, and these are endocrine disruptors. And I take an example which is very probably known to you here because it comes from a study on the Grand River. And what you see here, this nice fish, I learned to know just a couple of days ago, the rainbow darter, living in those streams. And the male, this is a male, they are very colorful. But if you have too much wastewater, unless it's properly treated, and I mentioned that afterwards, there's the problem for those fish that they actually have strong incidences of intersex. So what you see in the graph there on the y-axis, this is the incidence of intersex in those males, and on the x-axis you basically go down the Grand River from upstream of Waterloo wastewater treatment plant to downstream of, can I call it? Kitchener, sorry, thank you. The wastewater treatment plant. And what you see, there's a strong increase. There was a strong increase in the incidence of intersex due to chemicals discharged from the wastewater treatment plant. This has disappeared due to the upgrading of this Kitchener wastewater treatment plant. This was also in the daily news today. So this is just a kind of an example from the past showing, yes, those chemicals can have very strong biological effects. That's why we care about them. Now we've seen there's a large variety of those chemicals. They may be very potent with regard to biological effects. On the other hand, there are also treatment technologies that are able to remove a large fraction of those micropollutants out of wastewater, such as ozonation and powder activated carbon. And in Switzerland, it was decided a couple of years ago in a political process that a large fraction of the major wastewater treatment plants shall undergo an upgrading over the next couple of, basically 2 decades, to remove a large fraction of those micropollutants. It's not half of the wastewater treatment plants, sorry to correct Roy, it's 100 out of the 700, but they basically treat the largest fraction of the entire wastewater stream in Switzerland. One reason for that is also that with the current infrastructure that we have and that we are going to upgrade, it's also economically feasible to actually implement those additional treatment steps. Just to give you an example, it will cost every person in Switzerland about 3 to 4 coffee cups in Starbucks every year to actually finance that. And this was politically acceptable to say, yes, we would like to have insurance that we have also in the future good drinking water quality, and we do something good for the environmental quality in our streams, and we invest this money into upgrading. From a scientific point of view, actually, and this was then the starting point of this project Ecolmpact that Roy mentioned, we thought, okay, this is an interesting also opportunity for science to learn something about the ecological role of those micropollutants in streams. Because what is going to happen over the next years is that we carry out a kind of a real-world experiment in the sense that we will change the water quality in receiving streams in a rather defined manner upon the upgrading. And we thought we should take this opportunity and learn about the role of micropollutants in streams by actually observing how this change due to the upgrading is reflected in the stream ecosystems. And this actually then led to this project which basically followed these kind of overarching research questions. So we wanted to know whether the presence of micropollutants that are discharged with the wastewater have a significant influence on either the structure and or function of stream ecosystems. And if they did so, what were the underlying mechanisms causing these effects? So that's what we actually wanted to learn about over the last 4 years. Now, if you would like to identify the role of micropollutants in ecosystems, there are some challenges with that. So of course, many of the micropollutants are highly biologically active because they are actually designed to be so. For example, if you talk about pharmaceuticals or pesticides, they are designed to exert biological effects. On the other hand, despite what we measure in the stream, in general, these concentrations that we find in streams are the level where it's difficult to really see directly effects of a single compound where you can say, yes, I have this biological effect due to the presence of one particular chemical. The other point is that we have a large range of different chemicals, and they actually may affect different parts in the food web. So some of the chemicals, they may affect fish. Others may affect algae, plants, or invertebrates. So how is this what is the overall result then if you have this diversity of chemicals, but you also have the diversity of organisms living in those ecosystems? And then, of course, there are also many other influencing factors that act upon the stream ecology. If you just talk about wastewater itself, it's a mixture of many different things, not only micropollutants. Of course, you also have nutrients. You add heat to the stream, but you also have discharge of microorganisms, for example. So it's a whole set just if you look at wastewater itself. And then, of course, you have other influencing factors such as stream morphology, riparian vegetation that may differ. You have the hydrological regime, and so on. And there's a challenge how to actually tease apart the influence of all those factors and see what actually the micropollutants do to the stream ecosystems. We followed basically two research avenues to tackle those questions. And the first is actually the field part, the field survey, where we actually kind of try to implement this real-world experiment because we would like to see how those streams actually behave upon the upgrading of the wastewater treatment plant infrastructure. So this is the kind of the real-world part and the real-world experiment. And I will come to the experimental design, so to speak, with the next slide. On the other hand, it was also clear from the beginning that for certain questions, you would need to have more experimental freedom and actually the possibility, for example, to manipulate water quality to tease apart certain aspects. So, if something is not clear, just ask. So, okay, I will start with the field part. So basically, the experimental design that we have in mind for the entire real-world experiment consists of two temporal stages. And the first stage is actually to observe the baseline conditions, which means the conditions before the upgrading. And what we actually do is we compare at different sites the upstream and the downstream conditions at different wastewater treatment plants. So we can, by doing so, investigate what is the influence of the wastewater on those stream ecosystems under today's conditions. And if we have specific enough endpoints, we also can infer something about the role of micropollutants. And of course, it's the reference stage for the next stage, stage 2, where we then actually have different treatments after the infrastructure has been upgraded. So what we have are basically 4 different treatments that we can compare in the end. We have a status quo, so where nothing is going to change, so we run the treatment plants as they run today. Then we have 2 systems or 2 different technologies that go with the upgrading. So we have ozonation and we have powder activated carbon. And it depends on the specific localities where one of the technology will be implemented or not. And then we also have a force treatment, basically, which is actually the closing down of wastewater treatment plants, especially the smaller ones, where the wastewater will be then transferred to another plant. So this gives then a different kind of effects with regard to water quality, from no change to a complete reduction, for example, of the addition of nutrients and micropollutants if you have a shutdown. You have little changes for nutrient levels with regard to the upgrading, but you have about 80% reduction of the load of micropollutants. So these are then the treatments that we can observe in the future, so that we have, in a certain sense, what is called a Bachi design, so before, after, and control impact experimental design by properly selecting the sites that we investigate. Now, today, I basically can report on stage 1 because the upgrading has not been really implemented so far. So that's basically what I can tell you today. What is the up and downstream comparison and what do we learn out of that? So again, schematically, what we have the 2 stages, and on the y-axis, what I tried to show here schematically is a kind of a dissimilarity between the upstream and downstream situations today with regard to chemistry in the water, but also with regard to the biological situation. And of course, the hypothesis is that upon upgrading or removal of the wastewater, the dissimilarity between upstream and downstream situations will diminish. And the idea is that based on those changes, we actually can learn something about the role of micropollutants. Now, we try to implement this kind of experimental design in as many sites as possible across the Swiss plateau and the Jura Mountains. So you see here the dots indicating the sites that we actually included in the project. So we wanted to have many sites to get general results and not just results which are typical for one specific case. So we selected the sites in such a way that we didn't want to have any treated wastewater upstream of the wastewater treatment plant, and as much as possible wastewater downstream of the wastewater treatment plant, which means at least 20% of treated wastewater under low flow conditions. And then, of course, we also made sure that, let's say, stream morphology, riparian vegetation, and so on was as similar as possible between the upstream and the downstream locations. And if we apply these kind of selection criteria, we end up with about those 24 sites across Switzerland that you actually can include in the study. At each of those 24 sites, we implemented basically this kind of sampling and observation scheme. So you have the stream flowing this direction. You have here the discharge of the wastewater into the stream. And then we went downstream to such a distance that we had complete mixing of the discharged wastewater into the stream cross-section. So this basically defined very locally, very local specific, the distance to the downstream location. Then we had one upstream location as close as possible to the wastewater treatment plant. And then we added a second upstream location so that we actually have the possibility also in the future to observe what is the kind of the natural background variation with regard to different biological endpoints, for example, that may occur, that we can then compare with the upstream-downstream differences. So this was the layout that we had for the observations. And so at each of those locations then we carried out sampling for water quality, where we were measuring nutrients, general water quality, and then of course micropollutants. In the first set of the 12 sites, we used a large screening of about 450 compounds. We could then reduce that to 60 compounds in the second year because we noticed that we didn't lose much of information with this reduced set, but this allowed us to have a higher frequency of. And here just a few pictures of how these are mainly smaller streams, which is quite evident due to the selection criteria for the sites. Because if you avoid sites where you have already go to the uppermost wastewater treatment plant in the stream system, which then implies that you're basically. And now let's start to see some of the results that we obtained from this project and this field survey, water chemistry, because this is the kind of the driver that we actually are looking at with regard to its biological effects. So what you see here are different group of chemicals. So we start with, on the left is biologicals, other micropollutants such as household chemicals, but then also heavy metals and nutrients, for example, between the concentration measured downstream versus the concentration measured upstream. And what is clearly related to sewage, to wastewater treatment discharges, being pharmaceuticals, other household chemicals, the increase is the most pronounced if you look at the differences between upstream and downstream. So this is not really a surprise. But of course, we also have increases for pesticides, for example, and also for the nutrients. So they also play a role. The question is, what is the biological relevance of that? Now, a first way of actually looking at the potential biological relevance of those micropollutants as we have measured them in the streams is actually to take the measured concentrations and compare them to ecotoxicologically based so-called environmental quality standards and calculate the risk quotient for each of the compounds. And we used the suggested environmental quality standards for Switzerland as a metric to actually carry out this kind of risk assessment, and to obtain also kind of a very rough estimate of the total potential. We summed up those risk quotient for all of the compounds that we had. And there were two very interesting outcomes with this analysis. The first is that if we build this kind of risk quotient sums over all of the compounds, for all of the sites, in general there were, no, not in general, it was always the case that this risk sum was dominated by a few compounds. So initially we thought, okay, you have many compounds, so it will be distributed over many of them. Now it's a handful in general, between 5 and 10, that contribute 95 to 99% of the risk that we actually calculated. The other point that was also surprising to us is that the pesticides, although not being the dominant kind of pollutants with regard to concentrations, we're often driving the risk that we assess by this approach. And I come to that later on. And actually, if we go and take samples and carry out ecotox tests with the water from those different sites, we actually see how important pesticides can be. So if you, for example, look at how the water quality impairs the photosynthetic yield of algae, for example, you notice that the presence of wastewater actually has often quite a pronounced effect on the inhibition of photosynthetic activity. And this is shown here. What you see here on the y-axis, these are so-called diurone equivalent concentrations. So diurone is an important herbicide that inhibits the activity of photosystem 2. And you kind of normalize the activity of the entire mixture if you compare it to diurone equivalents. And what you see in blue, these are the values upstream of the wastewater treatment plant, and in green you see the corresponding value downstream. And in general, there's always an increase in the inhibition of photosynthesis activity downstream of the wastewater treatment plant, clearly showing, yes, there are herbicides in the wastewater that actually have an effect on an essential biological function. You may ask, what is about the estrogens that may be so important for fish? And we have seen the example how important that can be in the Grand River before the upgrading of the wastewater treatment plant. We also have yes tests, so tests that look for the estrogenicity in the water and also in the discharged wastewater. And this is then expressed in estradiol equivalent concentration, as you see them here. And here we have 3 bars. We have also measured the discharged wastewater with the orange bars, and then again blue and green for the upstream and downstream concentration. And in general, we really find very low levels of estrogenic activity. So this seems not to be a major problem anymore, given already the current kind of treatment, which removes a lot of those estrogens. And this fits very nicely together actually with the findings here from the Grand River. Yeah. Now you might say, okay, these are in vitro kind of assays. Do you also have results showing in situ in the real systems that we actually have effects on population, on communities, on ecological functions? And there are a number of results that actually can demonstrate these kind of effects in situ. And one of the very nice approaches to have a look at such effects is actually our tests carried out with periphyton, so the kind of the slimy thing living on stones and cobbles and so on in the stream bed, a mixture of algae, bacteria, fungi. So this periphyton can be used for so-called PICT tests, which stand for pollution-induced tolerance. What you do with those during those tests, you bring glass slides into the stream, and you let those organisms actually develop their biofilm on those glass slides. And you do that upstream and downstream of the respective wastewater treatment plant. At the same time, you deploy passive samplers, which specifically only sample the organic micropollutants in the stream water. And you do that at the same time. Then you bring back the biofilms to the lab, and you expose the biofilm to extracts from these passive samplers. So you basically expose the biofilms to the chemicals they have seen in the environment downstream of the wastewater treatment plant. So those chemicals that we have at the downstream locations. And then and this is shown here, you can, in a certain sense, make those response curves. So you measure certain biological endpoints, for example, bacterial production or photosynthetic yield, at different levels of concentration. And then you can compare whether or not the communities from upstream and downstream are sensitive to those chemicals to the same degree or not. And what we consistently observe is actually that the communities downstream, they are more tolerant. They stand more of those chemicals as compared to the community upstream. So there is an effect on the function of those communities. And this holds for algae and bacteria. We also observe changes between upstream and downstream if we look, for example, at macroinvertebrates and populations of them. What you see here are typical distributions of size distributions of gammarous amphipods. So these are important macroinvertebrates in stream ecosystems because they are strongly involved in the shredding, for example, of leaf material that falls into the stream. And what we consistently found is that upstream of the wastewater treatment plant, we have this pronounced peak of small individuals, big young individuals. And this peak is completely absent downstream of the wastewater treatment plants. So there seems to be a change in the structure of the population living upstream and downstream. Now, this was an effect on a single species. We were also interested in knowing what happens to the entire community of the macroinvertebrates in those streams. And of course, it's known for decades, if you have wastewater entering a stream, you will have effects on the macroinvertebrates. I mean, for example, the saprobic index was used for many decades already. Sorry. Showing the influence of wastewater, influence of organic material coming into the streams. So we wanted to have something which is a little bit more specific and may point actually to the toxicants that go with the wastewater. And therefore we used the SPEAR index as a metric for that point. SPEAR stands for species as risk with regard to pesticides in water. It was developed in Europe, originally in Germany, and then used now in many countries around the world to actually look at pesticide effects on macroinvertebrates. And what it actually does as an indicator, it classifies organisms, species, into two classes, either sensitive or insensitive towards pesticides, based on basically 5 life history traits of the organisms. And then you basically look at your composition of the community and the abundance, and then you basically look which fraction of the overall abundance consists of sensitive species versus non-sensitive species. And what we see in this graph, this needs a little bit of explanation. So from left to right, we have an increasing fraction of wastewater downstream of the treatment plants. And what you see here is the change of the SPEAR index from upstream to downstream at the respective locations. And the higher up we go, this indicates the higher the loss of sensitive species in the community. So also if you look at the macroinvertebrates as a community, we see a strong indication that due to toxic effects coming with the wastewater, we lose sensitive species. Finally, we were also interested in seeing whether or not there are effects on some relevant ecosystem functions. And again, I come back to leaf litter degradation. So this is an important process in many streams. And so what we did, we had leaf bags where we deployed defined amount, number, and a mass of dried leaves into the streams, upstream, downstream of the wastewater treatment plants. We let them in there for a couple of weeks, retrieved them, and then looked how much of this material was actually degraded and decomposed. And when you do that, you can then compile all of this data from the different sites. And what you see here is actually as a function on the x-axis of the nitrogen concentration of this leaf material. And one of the interesting things is, first, there's a kind of seems to be an optimum range with regard to the nutrients. So this is a kind of a hump-shaped curve, which was also observed some years ago in a broad study across Europe already, and our data nicely fit into what Guy Woodward and colleagues have published in Science some years ago. What is new here is that we now can compare how do the sites upstream and downstream of the wastewater treatment plant actually fall into this picture. And what you see here with the solid line, this is the kind of the envelope that encloses the points that go with the upstream locations. And the dashed line, this is the same for the downstream location. And what is now interesting, that even in the range where we have a kind of the optimal nutrient conditions, the degradation rate is always at a rather low level if we are at the downstream location. So there seems to be a toxic effect that limits the degradation rate of these leaf litter bags downstream of the wastewater treatment plants. And this is actually also shows up this kind of interplay between nutrients and toxicants. If we pull all of the data that we have on land use, on the wastewater fraction, on different aspects, nutrient levels, and micropollutant concentration in the stream, with, for example, the gammarous density that we find and actual leaf litter breakdown rates. So what we see is that certain constituents of the wastewater, like phosphorus or nitrogen, then they actually enhance the density of gammarids, which is then kind of enhancing also the degradation rate. On the other hand, we see that, for example, insecticides or other micropollutants, they have a negative effect. So we have oppositional influences of different wastewater constituents. So this might be actually one of the reasons why it's also difficult sometimes to actually tease apart and identify the role of micropollutants in stream ecosystems, because they co-occur with other constituents in wastewater which may have oppositional effects. Yeah, this is then the kind of the summary of some of those results that we have from the field. And the last point, so that we have these kind of opposing effects, actually renders itself to the conclusion that one would like to have experimental possibilities to actually work on better teasing apart the influence, for example, on nutrients and micropollutants. And in the remaining few minutes, I would show you the approach, how we try to follow that in a more experimental way. So we move away from the field and we go to a flume system that we developed in the course of this experiment or in this study. We call this flume system Maiandros, which is a Greek, I think, goddess or god for rivers. It consists of 16 different channels, flow-through channels, donut-shaped, but the water is circulating, but it has a residence time of about 12 minutes. And we can have 4 different treatments, so 4 different water quality flumes. And the flumes are, the treatments are then randomly attributed. And what you see in the background is, so this system is currently placed on an actual wastewater treatment plant. This gives us the possibility, for example, to run dilution experiments, water mixed in different ratios, and we can look at different biological endpoints. But we also have the possibility to actually just carry out spiking experiments, and that's what I would like to show you with one experimental result. So we actually designed a mixture of 17 different micropollutants, which normally occur in our streams. So we had a mixture of those 17 compounds, and we actually spiked that into the river water. And in one of the treatments, we also added then additionally nutrients. And in this case, we looked at degradation processes on cotton strip material. This is also, as leaf litter, quite a useful material to actually look at degradation processes. So what you see here is actually the mass loss after a certain time of exposure of these cotton strips in the different water qualities. We have 4 treatments. We have the control, which is basically the water from the river just flowing close by. We had to have a technical control, actually, which is just spiking a small amount of methanol into the water, because this amount of methanol was actually needed to also spike the micropollutants in the other treatments. So therefore, we had this kind of technical control. And then we had the spiking of micropollutants, and we had the spiking of the same mixture, the same concentration of micropollutants, but you also added nitrogen and phosphorus to the system. And then we were looking at how actually the cotton strips were degraded at which rate under those conditions. And what you see is that if you just add the 17 different micropollutants to the stream water, this has an influence on the degradation rate, which is lower than in the control. The other interesting point is, so there is a negative effect, but it disappears if you add additionally nitrogen and phosphorus to the system. So it seems this is a hypothesis that the heterotrophs that actually do the degradation, they have physiological costs first for detoxification because of the micropollutants, but they have also physiological costs that come with the acquisition of nutrients. And if you have a high nutrient level, though, those costs for nutrient acquisition might be lower, and therefore they can run degradation processes at a higher rate. With that, I'm already at the summary of those flume experiments. Still, I would like to point out one issue, one result. When we have those spiking experiments, and we measure the concentration in the flumes, the micropollutants, they seem not to be affected in their concentration. They basically are observed at the level that we spike them. Let's say, of course, we know we haven't measuring uncertainty, but let's say the influence of what happens to the chemicals while they are passing the flumes are less than what we actually can detect by our analytical techniques. And despite this kind of passing through the system without being affected, they exert ecological effects. So the majority of the mass of those micropollutants are just flushed through, but still the organisms get a sufficient high concentration into them to actually be biologically affected. And again, as in the field, we see the oppositional effects of nutrients and micropollutants. So with that, I'm coming again back to the research questions. Very briefly, we wanted to know whether there are effects on the structure of stream ecosystems. Yes, we see certain effects on the structure, for example, with the macroinvertebrates. What I did not show, but where we also have a clear indication is that, for example, diatoms respond in their community composition to the presence of micropollutants. But such effects might be partially masked by the presence of nutrients. And again, for the function, we also have seen that there are functional differences caused by the presence of micropollutants, such as induced tolerance. Where we have not made so much progress as we had hoped in the beginning is actually in understanding what are the actual mechanisms behind those effects that you observe on structure and function. So there's still hypotheses. What is the role of physiological adaptation? What is the role of ecological processes, such as species sorting? Or what might be the influence of evolutionary processes where you have for example, a change in frequency change in genotypes. So these are actually open questions that we would like to address in a follow-up project that where we are working on, as mentioned by Roy. One aspect that should also be mentioned here is that for certain of the effects, it might actually be that it's not the micropollutants, but it's the microorganisms coming from the wastewater treatment plants that actually may also play a role. So we also have strong indications for that. Okay, and this is then the outlook. So what we actually hope and what we have proposed to the Federal Office of the Environment, that actually stage 2 of this real-world experiment would be implemented in the framework of the National Monitoring Program of Rivers. And this is still in discussion, but I'm pretty positive that we will make at least a certain step into this direction so that this will be taken up. So that we actually can learn in the future upon the upgrading of wastewater treatment plants about their biological consequences in the streams. We also would like to support that by actually targeting, for example, master theses on specific sites where upgrading is taking place, that we actually can tackle those questions. As mentioned before, we are working on a follow-up proposal. It's also in the strategic planning of ERWAG to actually have a continuation of this program. So we will have also possibilities in the future for internal funding for running this interdisciplinary project. And in this context, so the idea is that we will have a core project that tackles a certain number of questions, and then we try to link other ongoing activities, like we have a project on antibiotic-resistant genes, we have a project on biotransformation that we actually will link to the future Ecompact Stage 2 project. With that, I would like to acknowledge first ERWAG for funding the core part of this project that I could present, also the Federal Office for the Environment. Of course, we had to collaborate, or not we had, we collaborated with many operators and on the wastewater treatment plants. They are very helpful, but they are also essential. You need to get access to those plants and everything. And then you see a large number of people doing a lot of work on different aspects. I can't name all of them, but I was speaking on behalf of them, and it was mostly great fun to work with them and also to work with them in the future. And with that, thank you for your attention.
We have time for questions. Simon.
Christian, that was wonderful. Thank you very much. The one thing we didn't talk about was fish. And is that because these are headwater streams where you don't find any fish?
No, it's no. You find fish there. So different fish species. Brown trout would be the most dominant one. So we had little activities on fish research, actually. We have some sites where we did fish studies. For example, we looked at gene expression with regard to detoxification. And it's actually interesting. This is the case where we have the situation, we have, on the one hand, we have a situation where actually wastewater was completely removed. And before that, there was a strong difference between upstream and downstream with regard to the expression of different genes that are related to detoxification of different kind of modes of action. And this kind of difference completely disappeared after the removal. And we also have first results on PICT assays on one side where actually powder activated carbon was implemented. And also the difference in tolerance between upstream and downstream has disappeared. But fish was for several reasons not in the main focus. One is just kind of operational, so fish move on large distances, so this makes it more difficult. Yeah. And it was, let's say, based on what we knew, also on the health of the fish, not, let's say, the main factor where we would say we should have a look at. Yeah.
Complicated study, interesting one. But in the field surveys,
Yeah.
comparison, but there's a lot of seasonality that goes on in these streams, a lot of flashiness of the hydrology and all these other things. So how did you standardize for when the samples are taken, or
Yeah.
how do you see beyond the seasonal?
So for example, the macroinvertebrates, they were always collected in spring. So we follow basically the biodiversity monitoring protocol of Switzerland. So that actually the data is directly comparable with other sites and other studies. And actually the timing depends then on altitude. So the lower the altitude, the earlier you start. Yeah, yeah. And for example, the leaf litter degradation assays, they were basically run in fall, so that we had, let's say, not too high temperatures, which then slows down the process a bit, then you have more time for exposure and so on. Yeah. But it's, of course, I mean, we have snapshots, so it's, this holds also for water quality. I mean, we have
deploying,
Sorry?
Deploying field crews out to get these things in place, kind of at the same time as Chuck.
Yeah, yeah, yeah. Yes?
Yes, it's an excellent presentation and a very important study in the context of the bigger thing that's been done there. Couple of questions. One is, are there many non-source inputs between the upstream and the downstream locations?
Mm-hmm.
The other one is that, of course, the wastewater discharge is going to just increase the general TOC, and different types of material perhaps provide variabilities. And is that somehow, are you somehow able to factor that out of the analyses that you're doing?
Mm-hmm. So the first thing is, we try to make sure that we have as little as possible other influences between the upstream and the downstream situations. So actually we had one case where it seems that we had a kind of a small point source for a herbicide, which might be the outlet of a farmyard or whatever. But in general, let's say the distances between upstream and downstream were short enough that, for example, diffuse pollution due to agriculture would not really differ. It differed, though, between the different sites. So if you look at so in a certain sense, this is also interesting because, let's say, the fraction of arable cropping upstream of the wastewater treatment plant range between 0 and 40% in the different catchments. So therefore you also see a difference in the kind of the background concentration of pesticides. So this then gives us the possibility to see how the input of wastewater kind of interacts with different degrees of pollution that come from upstream. With regard to the what we of course also see is that, for example, all the indicators for saprobicity also change between upstream and downstream. So there's a clear kind of a traditional signal of wastewater. Yes. Yeah. Yeah.
But is that increase in what we might call background organic matter from the wastewater, are you able to separate out the effect of that from the effects of the things that you're...
So for example, in this structural equation model where we look at the different potential influence factor, we take that into account. It seems not to be a driving factor that turns out to be relevant for these endpoints. Yeah. Yep.
Thank you. Can you comment on the impact of rainy season, dry season, and the concentration of pollutant and the outcome that you are reporting?
So what we tried to do was actually that we sampled for the water quality during dry flow conditions. So we completely left out rain events, discharge events, because they have, of course, a strong influence also on the background. So all the agricultural diffuse pollution pathways, they will be activated by rain events. So we avoided these kind of situations to have as comparable as possible situations with regard to sampling of water quality. But of course, this is an important aspect. So again, we have a snapshot. We have not covered the whole range of conditions that may prevail, especially with what happens upstream. There's also an issue with, let's say, rain-driven sources from urban areas, which we cannot completely avoid, like combined sewer overflows. We try to minimize the influence of that as far as possible, but go to Switzerland, it's very hard to find a situation where you really can avoid that. So there are, let's say, unknowns about these kind of rain-driven influences. But we try to tackle that, let's say, in a statistical manner by looking at the land use as a driving factor also in the statistical model. So looking how much percentage of urban areas versus arable cropping versus forest, and to see whether there's an influence of that. Apart from the percentage of arable cropping, we could not really find a land use characteristic that would show up strongly in the results.
But do you have data for different seasons?
For water quality, yes. Yeah. But we don't, we see some slight changes over the seasons, also with the micropollutants, but it's not tremendous and not that clear what it actually is. For example, for some pharmaceuticals, you have a decrease in summer. Is it just a change in consumption pattern? Is it that due to the higher temperature, the elimination efficiency is higher in the wastewater treatment plants? So there's questions open to that, yeah. But we have data across the seasons, yes.
Mark, you kind of answered my question. A lot of these streams are very pristine headwater streams. Did you see that your ability to detect the change decline if you looked at some of the more upstream impacted kind of sites? So if you look at sites that are more contaminated,
Yeah, yeah. I doubt, actually. So what we tried to do actually with the selection of the sites, we wanted to have a contrast between upstream and downstream as strong as possible. So no wastewater upstream and at least 20% downstream during low flow conditions. So this is the kind of the most extreme difference that we can get, let's say, under our conditions. And I would assume that once you have, if you go from 20% wastewater, or let's say on average flow 10% wastewater, then to 15% wastewater, I don't know or doubt that you would see a very strong signal. But one thing that we would have liked to do, but we had not resources to actually do it, to go further down at those sites and see how this is actually changing with distance. So this is an open question. This would be very interesting to see as well. Yeah, but we couldn't do that.
Great. One more question. So your wastewater fractions in your streams are generally pretty high compared to a lot of other places in sort of North America.
Mm-hmm.
So I guess do any of your studies kind of start to explore what level of dilution is a threshold between when you see effect? I know you have some model streams and so on. Did you look at any kind of, where is there a cutoff point where beyond which you don't see an effect?
No, I think one possibility to possibly address this question would actually be to take some of the, let's say, these structural equation models that come out of the analysis and kind of see by which degree you would need to, let's say, lower the input of a certain factor to actually still remain in the kind of the range, uncertain range of the undisturbed system, for example.
I was just sort of thinking about if you could take your results and extrapolate them to other streams.
Yeah. We didn't think about that so far, but it might be an idea to follow up. Yeah, thanks.
Is there one more question there?
Yeah, I had a quick question.
Yeah.
You mentioned this briefly. You talked about how you tried to sort of make sure that all the streams had similar morphology.
Mhm.
even within the same stream, you can have differences in habitat and population, I guess, makeup from like a pool and a riffle or whatever. So I was just wondering if you could maybe expand a little bit on how you controlled for that, or where you did your sampling.
I mean, if you do the sampling, for example, for the macroinvertebrates, you need to account for all of these microhabitats. So it's not just that you just sample riffles and not the rest. So there's a kind of a standardized procedure. Sorry for the standardized bill. But there is a procedure how to account for that, because of course you need to make sure that you have not a bias in what you actually sample. And this was also kind of considered when we actually went to the field and investigated the sites, whether they are suited or not, and whether the differences between upstream and downstream were kind of comparable. But of course, you always have also kind of a longitudinal kind of trend in such a system. You hardly can avoid that completely, but we selected only sites where, let's say, the basic microhabitats that are present in the streams were comparable upstream and downstream, also with the regard of their quantitative relevance. Yeah. Yeah.
Okay, I propose we stop here. Thanks very much, Christian, once again for your talk. Let's give him another hand. Christian is
the field and investigated the sites, whether they are suited or not, and whether the differences between upstream and downstream were kind of comparable. But of course, you always have also kind of a longitudinal kind of trend in such a system. You hardly can avoid that completely, but we selected only sites where, let's say, the basic microhabitats that are present in the streams were comparable upstream and downstream, also with the regard of their quantitative relevance. Yeah. Yeah.